and today I'm going to present my project carried out at the Institute of Ecology in UNAM, the National Autonomous University of Mexico City, titled Evolutionary History and the Importance of Genomic Diversity in the Face of Climate Change. As an object of study, we will focus on agaves. In general, all agave species can be considered keystone species in arid environments, as they offer abundant floral resources, such as nectar and pollen for pollinators. The genus includes over 200 species, with 100 of them being endemic to Mexico. Mexico is the center of origin and domestication of agaves. Agaves are native to the southern United States, Mexico, Central America, and the Caribbean islands. They prefer shaded shrublands, but can also be found in forest and tropical coastal environments. Agaves thrive at altitude ranging from sea level up to over 3,000 meters. They are relatively recent species, with a first divergence of 12 million years ago, related to the formation of arid environments on the continent, and the second of 3 million years ago, related to the floral reproductive systems and pollinators, mainly bats. In Mesoamerica, the use of agave plants dates back to pre-Hispanic times. In Tehuacan-Cuicuatlan Valley were found the oldest archaeological remains, estimated to be between 4,000 and 12,000 years ago. Indigenous people recognize these plants as a valuable source of raw materials and utilize them to produce various products including fibers, clothing, construction materials, food, beverage, and alcohols. Today, more than 70 traditional uses are recognized. In addition, agaves show great potential as bioengineered crops and contain the active compounds of medicinal importance since they have anti-cancer, anti-inflammatory, antioxidant, antimicrobial, antifungal, and probiotic properties. Agave marmorata is a semilparous long-lived species that attempts to flower for up to 30 years. Its main pollinator are bats. However, some species of birds such as calandrias and hummingbirds have also been reported in this plant. It is the species of agave with the most traditional uses, including the production of mezcal and gastronomy. They are diploid and genetic diversity have never been estimated. It is distributed in Puebla and Oaxaca in Mexico, in lowlands, forests, and shaded shrublands. The problematic with this species arise from the production of mezcal, which is one of the most significant uses of various agave species. This economic activity has the potential to drive development, particularly in the regions that produce mezcal, with a growing demand observed in recent years. According to Mezcal Regularity Council, mezcal contributed to 6 million pesos to the economy in 2019 and generates more than 20,000 direct jobs. However, mezcal production currently presents a sustainability challenge. While only a few agave species, particularly studied and cultivated species of mezcal producing, are still harvested from the wild. The rising global demand for mezcal has led to the exploitation of wild plants, displacement of traditional agave varieties, and deforestation for fuel. This demand has also increased the use of fertilizers and pesticides, resulting in erosion and environment contamination. Agaves are often harvested just before they flower, when they store the highest carbohydrates. This timing disrupts the reproduction and production of crucial nectar, pollen, and seeds vital for arid ecosystems. As a consequence, studies suggest that the mezcal boom is creating extinction depths in ecosystems, putting many species at extinction risk. Thus, the objective of this work was to estimate the genetic diversity and inbreeding of agave populations, studied using SNPs for conservation and management purposes. We also want to identify geographic patterns, management, genetic differentiation, and structure in all the populations. Also, we want to understand the demographic patterns through policy analysis and spatial distribution model. We perform spatial distribution models with different climate change scenarios. All these are for proposed conservation measures for each species and by management type. So, the methods in this species, the first one was we collected the different populations, we sent to sequence, then we filter for quality filters, we made a SNP calling with iParad, we estimated the genetic diversity, with R, and we made a spatial distribution model with biomodel. We also find the best fit for structured genomics, and we made coalescent analysis with fasting quad. So, in this species, we collect 4 types of management. The cultivated are the dark purple color, the light fence are the blue color, the nursery are the pink color, and the wild ones are the orange color. The genomic diversity and inbreeding estimated in this species was, with the expected heterozygosity and genomic diversity was 0.22. We also find 29,000 of SNPs, finding the greatest diversity in the wild population of San Juan Raya, followed by the light fence. In contrast, the locality that presented the lowest diversity was the cultivated ones of Santa Catarina Minas in Oaxaca. Regarding to the type of management, we found greater diversity in the wild population than in the cultivated. We also find a higher rate of inbreeding in cultivated plants than in wild ones. Regarding to the genomic structure with 2 different analyses, admixture and test, that take into account geographical coordinates, we found 2 genetic groups very marked by geographical issues. So we divide them into East and West, some of which are more similar to Puebla localities and the others to the Oaxaca. We find greater diversity in the Western group. We found a few localities with a mix of alleles that were wild from Puebla, and that also in the neighbor joining analysis present intermediate branch between the 2 groups. When doing the analysis by the management type, we did not find great differentiation, since all those cultivated were only found in the Oaxaca population. We made the historical demographics with passing qual. I test different evolution scenarios and will model a reduction, an expansion, and a reduction followed by an expansion. The most probable model was the last one, a reduction followed by an expansion, where I estimate a large effective ancestral population of 80,000 plants, followed by a contraction in the effective population size of 100 individuals, and then an expansion with effective population size greater than 8,000 in the last interglacial. The species distribution model I made 14 to analyze the trends in the population dynamics over time. I am only presenting 6 of them. Our findings indicate a significant reduction during the last interglacial period over 140 years ago, followed by an expansion during the last glacial maximum and another reduction during the Holocene. This demographic shift leads to the separation of the population, and they retreat into Pleistocene refuge, which promote independent evolution of the population. This process explains the pronounced population structure we observe today, consisting of 2 distinct genetic groups. Also, I calculate the future distribution of the habitat using an intense special SSP model that considers maximal fossil resource production and environment deterioration, and I found that there will be a habit reduction of -1 for the years 2041 and 2061. So, as conclusion, we found greater diversity in the wild than in cultivated ones. Geographical and demographic factors contribute to a genetic structure that is divided into eastern and western regions. Demographic analysis revealed a contraction during the Pleistocene, with low effective population size that promote independent evolution within the species. This was followed by a recent expansion characterized by a larger effective population size. We observe a reduction in habitat distribution in future climate change scenarios. As a conservation measure for cultivated localities, agroecological and agroforestry management is required, considering the types of soil and creating a soil recovery plan with crossbreeding and management programs. In wild population, it is essential to monitor genetic diversity and quantify the number of reproductive individuals. The use of nurseries should be encouraged to prevent the looting of seeds and plants. Additionally, when transplanting the F1 individuals to fragmented areas, it is important to consider a measure of genetic differentiation. So, thank you so much. If you have any question, I'm here. Thanks.